Okay, hello and welcome everybody. My name is Mia Raposa Peterson on the Biopharma Sales Executive team here at Oxford Nanopore Technologies. So thank you everybody for joining today's online session, which is going to be focused on resolving vector insertion sites and cell line characterization with long-read sequencing insights from print. I'm delighted to introduce Dr. Kyle Burbach, who you will hear from shortly. Kyle joins us from Addition Therapeutics, where he is a scientist too in molecular genome biology. Kyle received his PhD in molecular genetics and genomics from Washington University in St. Louis, where he studied Alzheimer's disease in the lab of Dr. Andrew Yu. At Addition Therapeutics, Kyle uses primarily long-read sequencing technologies to investigate the genomic safety of the print gene therapy platform. I'm going to start today with a quick overview of Oxford Nanopore and dive into a little bit of some big picture thinking of where we fit within the different areas within cell and gene therapy, as well as areas generally within your biopharma pipeline. And then we'll hand it over to Kyle. After all the talks are over, we'll also have a live Q&A session. So please add your questions today to the Q&A tab. We will try to get through as many questions as we can. The webinar today will be recorded and will be made available on demand after the broadcast. And with that, we will get started. So quickly again, not that we need to focus on this all too much, but just so that everybody knows what you'll be hearing from today, I'll kick it off. We'll go into a little bit of what it looks like in practice, and you'll hear from Kyle and again into Q&A. So I always really like to kick off these discussions with this slide. I really think that it does a nice job showcasing all the variety of areas within your pipeline where Nanopore can be a really valuable fit. So as you can see, we are supporting our pharma partners really all the way through their early R&D phases, all the way into their GMP manufacturing. So this has been a really exciting time at Oxford Nanopore as we really continue to expand our portfolio, all based on understanding the biopharma market needs and working together to answer your questions. So today we'll focus on a specific topic within insertion site analysis and Kyle's talk. But following the webinar today, we're always happy and love to meet with your team to learn more about the different areas of your research where we can help within these different application areas. I like to point this out just because Nanopore has really evolved in terms of the different support needs that are here to help you within these variety of different areas. So everybody here will have their own local commercial support as well as wet lab and bioinformatics teams to help you. So again, reach out to us and we'd love to hear from you. So before we dive into some of the details, I think it's always really important to kind of start where every therapeutic begins, which is understanding the underlying biology and with a bigger picture question. What's actually limiting success within drug development today? So we know that over about 90% of drug candidates actually fail. And a big part of that comes down to having incomplete molecular insight. So we're often making some really critical decisions based on having partial or fragmented data. And that challenge really continues to show up across the entirety of the pipeline, whether that's in your target discovery, biomarker identification, understanding mechanisms, or even within your quality control. So the question actually becomes, what if you could access a more complete view of biology, one that helps you to identify those better targets, uncover novel biomarkers, and understand that mechanism of action, and ultimately really make more confident decisions earlier on. So as therapeutic modalities continue to really become more and more complex, one of the biggest challenges that comes with that isn't simply just generating more sequencing data. It's actually having enough confidence to make the right biological and development decisions. So whether you're in your early discovery phase, analytical development, or manufacturing, the underlying question is actually very similar, which is, do we have enough information to confidently move the program forward? The specific questions evolve throughout development, but that objective stays the same, which is all around reducing uncertainty. So early in discovery, all the way to the left, the challenge is really, again, understanding biology. We want to understand things like transcript diversity, disease mechanisms, and ultimately, which biology is going to be most relevant to pursue. As programs mature, that focus shifts on understanding biology to understanding the actual therapeutic product itself. So now we're asking things like whether we're consistently producing the intended RNA molecule and whether we can comprehensively characterize its quality. Within your plasmid integrity, confidence in the RNA product also really depends on confidence in the DNA template that's used to generate it. So that raises questions around things like plasmid integrity, complete construct verification, and overall structural accuracy. And finally, for your engineered cell therapies, we really need confidence that genetic engineering occurred exactly as intended, including things like insertion sites, copy numbers, and structural integrity. So today, we're really going to focus on this section here of characterizing genomic integration. But again, all of these upfront steps are also extremely relevant and areas that we'd love to enable within your teams. This same theme actually continues to apply, and we really want to build confidence across pipeline, and it really applies still within your antibody development as well. So Nanopore can support questions, again, from your early candidate discovery through your library screening and through to your confirmation of that antibody construct. So importantly, for today's discussion, that doesn't necessarily stop once we have the right sequence. As that construct moves into a CHO production cell line, the question becomes, where and how did it integrate? And do we fully understand that resulting cell line? So the point here is to show that even across very different therapeutic workflows, the theme is again the same, reducing uncertainty as we move toward the final product. And I know there's probably a mix of audience here today in terms of levels of familiarity with the technology. But I like to show this slide because I find it really important to orient around how the technology works, since there are a lot of really key differences versus orthogonal methods that actually translate to a lot of our capabilities. So to quickly orient, the platform is built around our core chemistry made up of the nanopore, the motor enzyme, and then the run conditions that allow us to detect the flow of that current through the pore. As the molecules pass through the pore, this is what disrupts that ionic current. So we use then algorithms to interpret those changes in order to determine the current of that sequence. That electrical readout means that we can really carry out real-time base calling and analysis. So again, the way that this works really correlates to a lot of the benefits that come along with this method, especially when you're thinking about how you might use other orthogonal methods. So first, the sequencing happens all in real time. This really allows you to have real-time insights with the ability to potentially stop or start a run when you might have enough data. As we all can probably appreciate, time is money. Nanopore, for those here, again, might have varying levels of experiences, but we're typically known for long-read sequencing. And while that is absolutely true, we have made a lot of different improvements in terms of the flow cell architecture, the base calling algorithms, so that now you can actually detect things that are shorter fragments as well, down to about 50 to 100 base pairs. So this actually opens up a lot of different questions that you can continue to answer, again, all with one technology. And then again, some may know here, but we do offer a variety of sequencing devices really to match your scale. So this allows you to both explore the technology, but also have the ability to scale up with your high throughput project needs, both on the device side of things, but also within the different flow cell options that we have with our MinION and PromethION devices. And I won't go into that too much today, but again, we're happy to talk to you afterwards and understand what your needs are and advise accordingly. And lastly, a lot of our workflows really don't require you to perform PCR. So this allows you to maintain methylation information. Multiomics is always very valuable and again allows you to get all of this information all from one system, an effort for you to consolidate these different assays. So this theme can really be applied across a variety of different research questions and across your pipeline in order to consolidate and speed up the different areas of your development assays. So to transition into our main topic today, I also find it really useful to orient around what the community and publications are saying. So the community has actually identified a broader challenge across biological workflows, which is the reliability of downstream results is actually very closely tied to how well our starting materials are characterized. Studies that are looking at things like lab-made plasmids have found surprisingly high rates of sequencer design errors, while antibody research actually has faced very similar concerns around reagent identity and validation. The point here isn't necessarily the individual statistic. It's all around what happens next. If something is potentially wrong or poorly characterized early, that uncertainty doesn't disappear as you move downstream. So this can carry into experiments, engineered cell lines, and ultimately much more expensive development decisions. So why does this level of characterization really matter? All around because uncertainty has a cost. Across drug development, challenges around reproducibility and biological characterization have been estimated to carry a really significant economic impact in the tens of billions of dollars annually. But beyond the dollar figure, the real impact is what can mean for development, repeated work, delayed decisions, and potentially advancing biological systems that might not be fully characterized. And when we're actually engineering the genome, that last point becomes increasingly important. It's not necessarily enough to always ask whether the intended change occurred. Things like what actually happened in the genome, were there unexpected integrations, larger structural changes, and do we have enough genomic context to understand the complete event? As the complexity and the consequences of these engineered systems become more and more advanced, so does the importance that comes along with answering these questions with confidence. So another key thing to consider is the recent draft preclinical guidance from the FDA surrounding evolving expectations for sequencing-based safety evidence. To quote the guidance, it states that evaluation of longer stretches of sequence for the presence of large insertions or large deletions may require implementing a sequencing strategy that uses long-read sequencing-based methods that can detect such changes. So this is all a key piece to consider and something that we want to orient around, as our interpretation orients that these larger edits and rearrangements will be a really key piece where Nanopore can fit into answering your questions, but also within profiling the entirety of the different steps within your pipeline, again, all with one significant solution. So we've really spent a little bit of time here establishing the gaps in characterization and how they can have some real downstream consequences. But technically, I think we can ask the question of where does that risk come from? So our targeted and orthogonal methods can answer really specific questions. But by design, we're generally looking for something that you already expect to find. The risk can be what we didn't ask about, an unexpected integration, a rearrangement, or a genomic change outside of that targeted view. Short read sequencing gives us a much broader sequence information, but we're looking at potentially fragmented pieces of DNA. And as integrations become larger and again more complex, the risk can shift from detection to interpretation. So we may see evidence of an insertion or deletion, a rearrangement, or potentially multiple copies, but still really need to reconstruct how those pieces relate to one another and what that complete event really looks like. And again, this is all particularly important when we're making decisions around engineered cell lines or that therapeutic product. So what is the insertion site? Is the construct intact? How many copies are there and how are they arranged? So with Nanopore, the really useful advantage here is that long reads preserve more of that event all within the same molecule, allowing you to directly characterize that insertion together, all with its structural and genomic context, rather than having to heavily rely on inference. And just because the insertion is intact doesn't always necessarily mean that it's in the right location. Another key piece that you don't necessarily see here on the slide, but something that, again, we're always happy to talk more in depth with your teams about, is just considering things like overall turnaround time, CapEx needed, laboriousness of a workflow, and again, consolidation of assays, as you can think about a method and a technology to answer these different questions as you progress throughout your pipeline. So if Nanopore has the potential to give us the opportunity to reduce some of that uncertainty, the next question is likely, well, what's the right way to apply them? So the important point here is that there isn't really one approach for every insertion site analysis question. With Nanopore, we really have the advantage of being able to scale the strategy based on the level of characterization that's needed. So I won't go into too much detail here, just in an effort of time, but to summarize, on one end, we have our whole genome sequencing. This is always going to give you the most comprehensive view, and you'll hear more about this in practice from Kyle as we move on today. So with this approach, you're not necessarily having to restrict yourselves to predefined targets, so you can look really broadly across the genome for integration events while also gaining insight to things like structural variation, copy number changes, and other features. Our adaptive sampling, which I'll show you a quick example, but this is actually a really interesting middle ground and is a really unique and exciting opportunity within Nanopore. This actually allows you to enrich regions of interest without using PCR while still generating some background genomic information. So it's an opportunity to increase depth without having to really give up that broader context. And then for your really targeted questions, amplicon sequencing can provide a really rapid and scalable approach when we really kind of know what you might be looking for. For example, things like screening clones or confirming known integration sites. So as the question kind of moves towards your confirmation, towards comprehensive characterization, the strategy has an opportunity to move with it. So again, these aren't competing workflows, but it's all about matching the approach to the question and the level of risk that you're trying to address. So we'll wrap here quickly, but just want to show a little bit of an example and high-level overview of what adaptive sampling is, because it's a really, really unique and exciting advantage when you're thinking about Nanopore and how it can be applied to, again, answer different questions. So this is a really exciting method where people can explore the option for higher resolution insertion site analysis, and is really one of the unique capabilities, which is called adaptive sampling. So this allows you to selectively enrich for regions of interest, all done in real-time sequencing without having to do any additional wet lab, target enrichment, or redesigning of your workflow. So for applications like insertion site analysis, you can dynamically enrich for vector sequences or regions of interest all directly on the flow cell. The way that this works is actually where each molecule starts being sequenced, and the system will determine in real time whether it's on target or not, and it simply rejects it and moves on if it's not. So this will be based all within the design of your bed file that you would design for your targets of interest, or we do have some pre-designed panels as well. And from an operational standpoint, this actually simplifies the workflow pretty substantially. So reducing things like overall time, cost, and complexity, while also giving you just additional flexibility when you're thinking about adapting targets as your questions evolve. And again, we have a team of folks here on the bioinformatics side, on the wet lab side, all here to help support and help you optimize what this might look like to answer your questions. So to quickly show an example here, this is a publication called ASIS-seq. This is a really, really exciting use case of where Nanopore is being used within adaptive sampling. And what they did is actually where they designed a bed file to look at the vector sequences and then also the insertion event, so that they're able to get a low pass whole genome sequencing event, but then also really deeply characterize the areas that they might be looking at. And again, this is a way for them to be able to evolve their panels and the questions that they have as they continue to move on. So again, in an effort of time, I will absolutely am happy to share this paper with anybody who's interested, but wanted to flash this here just as an example of something that we're happy to explore with the teams further. So again, the examples that we've seen here all aim to highlight a bigger picture. What if a single platform could accelerate your discovery, improve your product understanding, and reduce risk all the way from your R&D through to manufacturing? So that's really the vision and overall theme behind Oxford Nanopore, providing one platform that really can deliver that value across every stage within your pipeline. So with that, I will hand the presentation over to Kyle. Please continue to add any questions using the Q&A button. And thank you all for your time. And Kyle, thank you for joining us, and I'll pass it over to you.
Yeah, thank you, Mia, and thank you to Oxford Nanopore for hosting me today. My name is Kyle Burbach. I'm a scientist at Addition Therapeutics, and today I'm going to be talking about my work using long-read whole genome sequencing to characterize the genomic safety of our PRINT gene therapy platform. Today I'm going to be talking briefly about what is PRINT, the value of whole genome long-read sequencing for our use case, some of the challenges that we've run into in aligning the transgenes that we're working with, as well as our target site in the rDNA and the solutions that we've come up with. And then finally, I'll talk about some of the actual data that we've generated. So current genetic medicines broadly fall into one of two camps. There are RNA therapeutics, which are very transient, they lack durability, but they tend to be very safe, very scalable, and have the feature of being redosable. On the other hand, you have gene therapies, which are very, very durable, but in general, they kind of fall short compared to RNA therapeutics in terms of safety, scalability, and often just redosability is not even an option. With PRINT, we're hoping to kind of combine the best of both worlds, create a highly durable medicine with genetic integration that is safe due to its RNA and LNP-based delivery, highly scalable, and redosable when needed. So what is PRINT? PRINT stands for precise RNA-mediated insertion of transgenes. And what we do is we take two RNA molecules and package them in an LNP. One of these RNAs is an mRNA, which codes for the R2 retrotransposon, which finds its template RNA, which is the other RNA we deliver, through a structural element on the molecule. They form an RNP and they migrate into the nucleus. Once there, R2 finds its target site in the rDNA, forms a single-stranded nick, and then uses that flap to serve as a primer and start cDNA synthesis using the RNA as a template. From there, once it has its full cDNA synthesized, it will do a second strand nick and second strand synthesis. And once those ends are repaired, you have your full complete gene insertion into the target site for the in the rDNA. And so when we want to assay, is this transgene ending up where we want it to be, there's several methods that we can choose from and we considered when we started this. LAM-PCR is a cost-effective, sensitive method that's widely used, it also benefits in that you don't need to modify your transgene at all to use. However, it comes with a lot of bias, both in the preparation of the material through restriction enzyme or shearing bias, as well as in the preparation of the library. You go through altogether 3 rounds of PCR to get your final library. It also suffers from some PCR-based artifacts because of that. It's not very specific as a result. And because it's a short read method that assays off of one end, you don't get full information about your transgene. A method that's kind of in development right now that's pretty exciting for us is called RippleSeq. It involves putting a promoter in your transgene that you can use to do in vitro transcription and cDNA synthesis and use that to generate your library. It's very cost-effective and very straightforward. It's sensitive and specific, and depending on how you design the assay, it can be extremely informative. On the downside, it's a new method, so it's not widely used. It does require modifications to your transgene with the addition of this promoter, and so it only can really serve as a surrogate assay and therefore is not clinically relevant. And so that kind of leads us to long-read whole genome sequencing, which was kind of my method of choice when we got started because it's very unbiased, there's no PCR, simple, specific, doesn't require modifications to your transgene, and it gives you full molecule information. On top of that, beyond the transgene, it also can give you whole genome information, which can be used to assay methylation of sites of interest, as well as whole genome stability. On the other hand, if all you're interested in is your transgenes, in that case, most of your reads are going to be uninformative. It also tends to be less sensitive than other methods, and therefore ends up being a little bit more expensive to acquire the depth of sequencing that you need to get high confidence data. And so with our transgene, we discovered quickly there were kind of 2 problems we were going to run into. One is that with a lot of immortalized cell lines, some of the sequences that are used to accomplish that are similar to sequences that are commonly used as regulatory elements in kind of gene therapy development. And so that can lead to frivolous off-target mappings. The other one is when we were kind of designing test data with R2 as a non-LTR based retrotransposon. It's known that they can result in kind of truncated insertions from the 3' to the 5' end. So what we really want is this top example here where you get full addition of your transgene. However, we also expect to, a certain amount of time, get these truncated events where some of them will be functional as long as they get the promoter and the transgene. Some of them will not be functional because they're missing too much cargo. We also wanted to be very thorough in kind of checking off all the possibilities. And so we wanted to make sure that, any kind of integration event was accounted for. And what we found in our test data was that these kind of bottom 2, the deletion event and the insertion event, actually posed a little bit of a challenge for Minimap2, where it only really successfully maps 1 bit of the sequence and therefore mislabels the event. And so we kind of have 2 problems. Complex insertion events can be mislabeled, and off-targets are inflated by contaminant alignment. And I found a single solution that answers both of these problems, which is to take our transgene reference and actually break it into pieces. This allows us to more sensitively align the transgene to small pieces that might be integrated, solving the complex insertion event problem. And by breaking it apart like this, we're also able to remove any sequence elements that might be present in cell lines that we're using, and therefore avoid off-target mapping that we wouldn't want. And so our transgene alignment pipeline ends up looking like this, where we take our raw FASTQ, align it against our chunked reference, and use that to get a list of reads containing transgenes. We can extract specifically those so that we don't get any contaminant reads, and then align it against the full transgene. And like I said, this will sometimes miss some of the structural information, but by comparing it back to our original chunked alignment, we're able to resolve those more sensitively. And we get accurate mapping in a pure transgene population, which is really, really good. The other problem we have is that R2's target site is in a sequence called the rDNA, which is the ribosomal DNA. And if you look at this karyotype I'm presenting here, you can see these blue regions on chromosomes 13, 14, 15, 21, and 22. These are called the nucleolar organizing regions, and they are the home of the rDNA. And you can kind of see structurally how that looks on the right, where in between the very, very end of the chromosome, which is the distal junction, and the more centromeric region, which is the proximal junction, we have many, many repeating units of rDNA. Per NOR, it's somewhere between 20 and 200. Each of these units is about 44 kilobases long. And so per chromosome, we're looking at least a megabase, easily upwards of sequence that is highly, highly repetitive. So this has historically been a real challenge to resolve in genome assemblies. And even in the recent T-to-T assembly, which is arguably one of our most complete assemblies to date, they don't actually sequence through the entire nucleolar organizing region. Here you can see the structure of a single rDNA repeat, and you'll see that only about a third of this 44 kilobases is actually coding in regulatory sequence. The target site for R2 is right here in the 28S coding sequence, and typically we're adding at least a kilobase, usually more, and so short read sequencing is not able to give us full molecule information here, and so long read sequencing is really powerful to be able to get at this, especially as we start working on longer and longer cargos. Additionally, when looking at this karyotype, you can see that there are some regions I've highlighted in pink, and these represent duplications of R2's specific target site throughout the genome. There are at least 12 well-preserved duplications of this target site, and most of them have homology longer than the read length for short read sequencing. And what we experienced is that trying to use short read sequencing resulted in most of our reads actually being labeled off target, because when multi-mapping occurs, it's going to pick somewhat randomly which site to assign it to. And so many of our sites were being many of our transgenes were being mapped to one of these sites and being labeled off target. But with long read sequencing, we're actually able to extend past this maximum 1,200 base pairs of homology and accurately map to either the rDNA specifically or one of these target sites. And so when aligning to the entire genome, our flanking sequences to map on and off targets, we run into a couple of challenges. Because of the length of the nucleolar organizing regions, we cannot identify specific rDNA units. And so we end up with a lot of multi-mapping, especially with these duplicated regions. It can decrease the map quality from Minimap. And so what I ended up doing was actually masking out these p-arms entirely and supplementing with a single unit of the rDNA. Having this single rDNA unit to align against provides a lot more confidence in the mapping and results in kind of better calls downstream. And so now that we have kind of all of the kinks worked out of our pipeline, here's what it ends up looking like. I showed you the transgene alignment earlier, but then we can use that information to get the flanking sequences as a FASTQ and align that against our modified HG38 to determine our on and off target mapping. We also take this same modified HG38 reference to do whole genome mapping of our raw FASTQ to look at things like methylation, ploidy, instability, etc. And all of the tools that I've used are here on the right. And so now I can show you some actual data. This was an experiment we did looking at different template architectures for our template RNA. And what I want to remind you is that R2 is a non-LTR retrotransposon, so it writes 3' to 5', resulting in early termination, which we call truncations. And what we're measuring here is the percent of reads that actually make it past the promoter and are therefore functional when integrated into the genome. And what you can see is that just by changing the sequence composition of the template, we're able to get massive differences in the percent of reads that integrate into the or that reach functional length before integrating into the genome, anywhere from 6% on our worst performing template to almost 35% on our best performing template. And this is information that you can get by doing ddPCR, by counting the number of events at the 3' end and the 5' end and just doing that division. However, what's really useful about long-read sequencing is that it gives us information about the entire length of the transgene. And we can see which specific sequence elements are performing better or worse and use that information to rationally develop our template in future design iterations. And that's proven to be a really, really useful tool. Furthermore, when we're looking at actual targeting of the integration, here you can see how that breaks down in some clinically relevant cell types. On the left, we have data from primary human hepatocytes that we printed, and on the right are primary human T cells. And what you can see is that in both of these clinically relevant cell types, over 99% of our events are mapping directly to the rDNA. And this is really good and is only possible thanks to the long-read sequencing that we're taking advantage of. Additionally, we can see that this less than 1% that are not mapping to the rDNA are actually mapping to the site that we are aware of from our bioinformatics predictions, one of these target site duplications that exist throughout the genome, leaving us with so far 0 off targets at random unintended sites in the genome. That being said, our sequencing depth for the hepatocytes is just over 500. Our limit of detection with 95% confidence on that is about 0.5%. And so we're not trying to claim that it never, ever hits off targets, but so far the data is pretty compelling that it is going to be a very low percentage of the time. The T cells, obviously, this is the 95% confidence interval is a little bit higher, but we are doing additional sequencing to raise our N on that. And so this is really, really good data that is made better through the use of long-read sequencing. And finally, outside of the transgene, we chose to do long-read whole genome sequencing because of the additional information that we can get from it. So on the left, you can see a plot showing the average methylation of the rDNA across the hundreds of copies that exist per genome. And this shows us basically in the target site, which is in the dotted line in the coding sequence of the rDNA highlighted in yellow, how many or what proportion of rDNA units are being actively transcribed, which from a rDNA biology perspective, an rDNA genomics perspective, is really interesting information because there's a lot of discussion about there are different numbers of rDNA units per cell, per tissue, per individual. So seeing how many are being actively transcribed in relation to how many actually exist in the genome is really useful information. Additionally, despite decades of studying the rDNA, the promoter has remained elusive. And with this methylation data, we can actually see, I predicted just conservatively that maybe it's about 2 kb upstream of the coding sequence. And so that's where the yellow highlighting ends. But you can actually see it's probably less than that based on the methylation pattern here. So personally, as someone with a background in genomics, I think that's really, really cool. Additionally, we're able to look at the kind of bulk karyotype of our cells. This karyotype I'm showing here is from RPE1s, which are an immortalized retinal pigment cell type. And what you can see is that this whole segment of chromosome 10 is actually triploid. This is something that we had a loose inkling of before doing this, because ddPCR assays had shown a discrepancy between one of the reference genes that sits in this region versus the other ones. And I actually developed this karyotype about a year before a reference genome for RP1s was published, which confirmed this trisomy of this region. It's actually attached to the end of chromosome X. And so for other cell lines that don't have reference genomes published, we can do whole genome sequencing, learn this information, and use that to inform other assays that we're doing in-house that are important for our QC and validation and everything. Furthermore, we are currently developing a pipeline to look at structural variation. This has proven a little bit challenging because we're trying to make it more sensitive to be able to detect events that might be occurring in single cells or in small clonal populations. There's a lot of heterogeneity that occurs between cells in a dish when you expose them all to print. So we're working on that actively. And then we're also trying to look at per read methylation to get at a little bit of what are the accessibility requirements for the R2 retrotransposon. So in conclusion, I hope I've demonstrated that whole genome long-read sequencing is a very powerful tool for our use case of investigating transgene insertion via print. I really like it because it is probably the method that introduces the least bias in the preparation and sequencing. That being said, obviously there's always going to be a trade-off, and in this case, that's cost and sensitivity. It ends up being a little bit more expensive per transgene compared to methods where you do targeting or enrichment. Picking the right tool for the job matters a lot, and in a lot of cases, this adaptive sampling tool that exists for Nanopore might be able to get around that. We chose not to use it because we wanted to make sure that we got at least 2 kilobases of flanking sequence on either side of our transgene. And I think when I was talking to the technical supports a couple years ago, they said it's about 500 base pairs that it'll read before it makes a decision. Furthermore, biology matters, and biology matters to computational biology as well, which is something I think can get forgotten a lot. Pipelines aren't one size fits all. Commonly used regulatory sequences in gene editing and synthetic biology cause conflict, and that has to be accounted for. Otherwise, you get thousands of apparent off targets in your data. Furthermore, the rDNA specifically is a complicated piece of the genome, and it really requires some dedicated workarounds to be successful in its mapping and its processing. And as sequencing technologies improve, hopefully that will improve as well. Moving forward, addition, taking the improvements that we have made and that we're currently working on, we're advancing pipelines in Fabry's disease and hypothalamic obesity. And we have some pretty compelling data supporting other programs that we are pursuing with partners. And so with that, I'd like to thank Oxford Nanopore for giving me the time. I'd like to thank all of you for attending.
So again, thank you so much, Kyle, for your presentation. And thank you to everybody who has joined us here online. Just to wrap up, you can find more information about Oxford Nanopore Biopharma Solutions using the QR code that you can find here on the screen. And if you have further questions about your projects or getting started, you can always email us at support@nanoporetech.com and we'll get you in contact with the right team member. So thank you all. And I think there is one more slide. Yes, our quick disclaimer. This is our standard disclaimer. Oxford Nanopore products are currently being used for RUO. And that is all for today. Thank you all so much for joining us. And we hope to see you soon. Thank you.